Hello everyone, I'm Sarveen, a third-year biological sciences student from Dr. Wee-Young's lab, and this summer I explored Lumican, a structural protein in the brain, to understand its role in multiple sclerosis and demyelinating models. Multiple sclerosis is a chronic demyelinating disease that is characterized by inflammatory lesions that damage the brain, optic nerve, and spinal cord. This leads to the loss of myelin, the coating that helps nerve signals travel quickly, and when myelin is damaged, communication between nerve cells becomes disrupted. These changes give rise to symptoms such as vision problems, fatigue, cognitive difficulties, and motor impairment. MS usually begins with periods of relapse and recovery, but over time the damage becomes progressive and harder to treat, making it one of the biggest challenges in MS research today. One of the features of MS lesions is changes to the extracellular matrix, or the ECM, which is a complex network of molecules found throughout the extracellular space of tissues. In MS, ECM molecules such as degeneration can accumulate around blood vessels, which can influence immune cell entry, and others such as fibrillin 2 can accumulate within the tissue, where they may impair remyelination. And even though some ECM molecules in MS are known, the full landscape is unclear. To address this, our lab did proteomic work to identify Lumican as a new ECM molecule in MS tissue. Lumican belongs to the SLRP family, and it helps to organize collagen and other matrix proteins. It's normally found in fibrous tissues like the cornea, tendons, and meninges, but its role in the brain remains unknown. Our data show that it's mainly produced by fibroblast-like cells, so my research question is Lumican expression elevated in MS, and could it be linked to failed repair? And I hypothesized that Lumican increases in MS lesions and in its experimental demyelinating models. My three aims are firstly to quantify Lumican in MS lesions, secondly to measure Lumican in two demyelinating models, and thirdly to identify which cells produce it. To address these aims, I first spent a lot of time optimizing and testing antibodies until we were finally able to clearly visualize Lumican in these lesions. After that, I scanned the slides on the slide scanner and did my analysis in QPath to detect positive cells and perform cell counts, and then ran statistical analysis in GraphPad. Here I am showing the three types of tissue I analyzed: the control white matter, active lesions, and chronic active lesions. Control white matter is healthy brain tissue from people without MS. Active lesions are areas of strong inflammation and ongoing myelin loss, while chronic active lesions have inflammation mainly around the rim. Now for the results, when we stained the MS tissues for Lumican, shown here in green, we found that its signal was much stronger in active and chronic active lesions compared to control tissue. It appeared mainly around blood vessels, which are regions linked to inflammation, scarring, and poor tissue repair. Next, we looked at cells which produce Lumican. By combining Lumican staining with fibroblast markers, PDGFR-beta and collagen 1A1, we found a significant increase in triple positive cells in both active and chronic active lesions compared to control white matter. We then also looked at animal models to see how Lumican changes at different time points during demyelination. The first model, EAE, progresses from healthy control, which is called naive CFA, to a chronic stage where inflammation decreases, but repair has failed. Across these stages, Lumican increased and was highest in the chronic stage. The next model, lysolecithin, involves injecting a detergent into the spinal cord of a mouse to create a small demyelinated lesion. And across days 3, 10, and 14, Lumican gradually increased inside the fibrotic core, which is an area with little to no repair. Together, these results show that Lumican-positive fibroblast-like cells are increased in active and chronic active MS lesions, mainly around blood vessels. And in both demyelinating models, the EAE and lysolecithin model, we see that Lumican levels also increase over time, especially at stages where remyelination fails. Overall, Lumican seems to be linked to this non-repair environment, but whether it actively drives failed repair is still unknown and will require future experiments. For example, testing what happens to remyelination when Lumican is removed in mouse models. I'd like to thank Dr. Wee-Young, Dr. Rianne Gorder, my supervisor, and our lab members, as well as the Alberta MS Network for their support and funding. Thank you.